Have you ever noticed how some 70-year-olds are out there running marathons while some 50-year-olds struggle to just, get up the stairs?
Right. The numbers on our birth certificates are lying to us.
Exactly. They do not match the biological reality of our bodies. So today, we are going to do a deep dive to unpack the exact biological clocks ticking inside you.
And crucially, how your daily lifestyle might be actively speeding them up or slowing them down.
Yeah, so our primary source today is this absolute landmark 2026 study published in the journal Cell. It's by Lai and colleagues, and it details something called the X-age project.
Right, specifically looking at the mCAS cohort. That stands for the multicentric Chinese aging standardized cohort.
And the scale of this is just, it's staggering. They tracked over 2,000 individuals across 10 different biological dimensions.
Yeah, generating over 1 billion high-quality data points, which is, unprecedented.
A billion data points. So the mission for today's deep dive is to take that high-definition molecular map and figure out exactly how our organs age. Because, spoiler alert, they age at completely different speeds.
Oh, absolutely. And we'll also uncover this massive plot twist in the data about what is mechanically pushing the aging process forward.
Right. So let's just jump in. Let's start with the visible surface of aging. The researchers call this the core capacity clock, or the CC clock.
Yeah, this is essentially the outside-in view. They measured 240 different parameters across 8 physical modules.
So we're talking about things like body morphology, your cardiovascular system, respiratory, metabolic, immune, endocrine, motor, and neurocognitive functions.
Exactly. They translated all that visible and functional decline into a highly quantifiable, mathematical score.
Like, give me an example. What's a visible marker?
Well, using digital modeling, they found that just the ratio of facial wrinkles alone, just wrinkles, had a 93% predictive accuracy for a person's age.
Wow, 93% just from wrinkles.
Yeah. And they also measured central obesity, tracking waist-to-hip ratios, and they looked at declining bone density.
Okay, but those are static things. What about functional tests?
For motor skills, they looked at grip strength, like how hard you can squeeze a device, and the single-leg stance, basically how long you can balance on one foot.
Oh, I've tried that one. It's harder than it looks.
It really is. And for respiratory health, they measured something called FEV1, that's forced expiratory volume, which tracks how much air your lungs can forcefully expel in exactly 1 second.
Right. And they even had people do cognitive puzzles like the trail making test.
Yeah, where you connect dots in a specific alternating sequence under time pressure. It measures executive function and visual attention.
But, okay, here is where I have to push back a little on the premise of this whole CC clock.
Sure.
We've known for centuries that as humans get older, they get wrinkles, they walk slower, and their balance gets worse. My grandma could have told me that. What is scientifically revolutionary about putting these basic tests into a clock?
That's a fair question. The revolution here is the mathematical integration.
Meaning what exactly?
The CC clock takes highly diverse signals, from the physical degradation of your skin to the processing speed of your brain's white matter, and synthesizes them into a single objective score of physiological capacity.
Oh, I see.
It proves mathematically that human aging isn't just, chaotic wear and tear. It follows a highly predictable structural decline.
Like a car dashboard, you're checking the tire pressure, the engine light, the oil levels, and rolling them into one overall vehicle health score.
Exactly.
Okay, let's do a quick mini recap of this first section before we move on, just to lock it in.
Good idea.
So bullet point 1. The CC clock measures 8 bodily functions to determine your physical biological age.
Right. And bullet point 2. Simple things like your physical balance and grip strength are major mathematically validated indicators of this structural decline.
So question for you listening right now. If you stood up and tried the single leg stance test today, what age do you think your balance would actually reveal?
It's a sobering thought for sure.
Definitely. But the CC clock only measures the downstream effects, right? The visible damage.
Exactly. To understand the root causes, the researchers had to look under the biological hood at 6 different molecular layers. They created what they call the multi-omics clocks.
Omics? That's a heavy science word. It basically just refers to comprehensive mapping, right?
Yes. They mapped the DNA methylome, the transcriptome, the proteome, the metabolome, and the gut microbiome.
Okay, let's break those down. Let's start with the DNA methylome because the study highlighted this as the most highly predictive layer.
Right. So think of your DNA as a massive, ancient recipe book for building you.
Okay, a recipe book.
Methylation is like sticking read or do not read Post-it notes on specific pages of that book.
Oh, I love that analogy.
Yeah, the recipes themselves, your underlying genes, don't change. But which ones the cellular chef actually decides to cook? Well, that changes over time based on those Post-it notes.
So as we age, our bodies stick those do not read notes on the good genes that maintain health, and they pull the notes off the bad genes that trigger inflammation.
You've got it perfectly. And the study found a specific marker on a gene called ELVL2.
ELOVL2, what does that one do?
It helps manage lipid synthesis, basically how your body processes fats. And the methylation on that specific gene is the most accurate single molecular predictor of chronological age out of everything they tested.
Wow. Just checking the Post-it note on the fat processing gene tells you how old someone is.
Pretty much. But the DNA tags are just the instructions. We also have to look at what the cells are actually doing.
Right, which brings us to the transcriptome and the proteome.
Exactly. The transcriptome is the messenger RNA. That's the courier carrying those DNA recipes out to the protein factories.
And the proteome is the actual proteins doing the work.
Right. While DNA methylation is great for predicting chronological age, your active proteins and your metabolites, the metabolome show the most dynamic changes related to how fast you are biologically aging.
Metabolites being the chemical leftovers, right? Like the exhaust from your cellular engines.
Exactly. And in the proteome, the researchers highlighted a specific protein called CILP2. It's an extracellular matrix protein.
And what happens when CILP2 builds up?
As it accumulates, it actively degrades joint cartilage.
Ah, so that's the creaky knees protein.
That's a great way to put it, yeah. The creaky knees protein. And then in the metabolome layer, they found elevated levels of cysteine.
Cysteine, what does that indicate?
It indicates high oxidative stress. Your cells are essentially rusting from the inside out because that metabolic exhaust isn't being cleared efficiently.
Okay, wait. So they have physical data, DNA tags, RNA couriers, active proteins, cellular exhaust, plus the gut microbiome, which is trillions of bacteria. How do you make sense of all that without your brain exploding?
You use advanced AI, specifically architectures featuring two distinct mechanisms, attention and inception.
Okay, AI always sounds like a black box to me. How do attention and inception actually read biological age?
It's fascinating. The attention mechanism acts like a spotlight. It dynamically weighs the different omics layers depending on the specific trait it's analyzing.
So if it's looking at, say, joint degradation, it shines the spotlight heavily on the proteome to look for that CILP2 protein.
Exactly, you nailed it. And then the inception mechanism allows the AI to process multiple streams of data at different scales simultaneously.
So it overlays them.
Right. It sees how a faulty DNA tag creates a flawed RNA messenger, which builds a damaged protein, which then results in toxic metabolic exhaust. It captures the entire causal chain.
That is incredible. And this AI builds the multimodal clock, or clock, which completely outperforms any single test.
It's the ultimate biological read.
Let's do our second quick recap here.
Sure.
Bullet 1. Omics means mapping your molecules from the Post-it notes on your DNA to the active proteins degrading your joints.
And bullet 2. The clock uses AI to combine your physical tests with this deep molecular data for a perfect biological age read.
So question to you listening. If you mapped your internal molecular clocks right now, which one is ticking the fastest? Are you accumulating joint-degrading proteins, or are your cells drowning in oxidative rust?
And that concept of different clocks ticking at different speeds is crucial because it shatters the illusion that you are aging as a single uniform entity.
Right. I'm not just aging, my liver is aging, my heart is aging, my skin is aging.
Exactly. The researchers built specific clocks for 6 different organs and found severe organ asynchrony, meaning they don't age together.
What's the canary in the coal mine? Which organ goes first?
The liver. It's essentially the body's primary filtration plant, and it hits a massive aging inflection point as early as age 40.
Age 40, that seems incredibly early for a major organ to just start failing its inspections.
Well, the liver does the heavy lifting. It manages lipid metabolism and filters toxins. When those filters get clogged in your 40s, the toxins and inflammatory markers spill over into the bloodstream.
Oh, man. So it poisons the well.
Right. And that systemic spillover is exactly why the brain, which is highly sensitive to inflammation and sits downstream from that filtration plant, takes a major hit a full decade later. The brain's primary aging cliff hits around age 50.
But wait, how do they actually track this? Like, they obviously aren't taking liver and brain biopsies from 2,000 healthy people just to see how old their organs are.
No, definitely not. They identified what they call proxy clocks.
Proxy clocks, how do those work?
By analyzing the blood, they found a specific panel of about 100 proteins that act as a highly accurate proxy to predict the health of these internal organs.
So the organs leak specific proteins into the blood as they break down.
Exactly. Testing those leaked proteins tells the whole story without needing a single biopsy.
Which means we could soon have a future where your annual physical doesn't just check your cholesterol. A doctor could literally tell you, hey, your liver is 45, but your brain is 35.
Yes. And we desperately need that early warning system because, as the data shows, human aging isn't a slow, steady slope. We fall off cliffs.
Yeah, let's talk about the two cliffs of systemic aging. Cliff 1, you said, happens between age 40 and 50.
Right. This period is marked heavily by hormonal fluctuations and lipid, or fat, accumulation, which perfectly aligns with that early liver aging we just discussed.
Makes sense. And then there's Cliff 2, hitting between age 60 and 70.
That cliff is characterized by dropping bone density, increased blood pressure, and this is huge, a massive spike in blood coagulation factors.
Coagulation, like blood clotting. We are definitely going to come back to that spike because I know that is the big plot twist of this deep dive.
Oh, it completely changes everything we know about aging.
Okay, time for our third recap. Bullet 1. Organs age at entirely different speeds. Your liver generally ages a full decade earlier than your brain.
And bullet 2. Aging accelerates heavily at two distinct periods. The hormone and lipid cliff in your 40s, and the bone density and blood pressure cliff in your 60s.
So listener, think about the people you know in their late 40s or late 60s. Have you noticed these sudden cliffs in their overall vitality?
It's usually pretty visible once you know what to look for.
Definitely. Okay, so we know the cliffs exist, but what pushes us over the edge faster? And more importantly, what keeps us away from the edge?
Right. The researchers measured the gap between chronological age and biological age to identify fast agers versus slow agers.
Let's start with the fast agers. What are they doing wrong?
The data is pretty stark. Heavy smoking, for instance, adds roughly 4 years to a person's biological lung age.
4 years, just gone.
Yeah. And short sleep, meaning consistently getting under 6 hours a night along with high meal frequency, also correlated strongly with accelerated molecular breakdown.
And physically, these fast agers had higher blood pressure and wider waist-to-hip ratios, right?
Exactly. Their internal multi-omics clocks were basically sprinting.
Okay, but I want to be a slow ager.
Oh.
How do we slow down our molecular decay? The source material had this great mnemonic that I want to share.
Oh, the swag mnemonic.
Yes! Swag. S-W-A-G. Let's break it down. The S is for sleep, but it's not just how much you sleep, it's the timing, right?
This is fascinating. The study showed an optimal bedtime of 2,100 to 2,300 hours.
So going to sleep between 9 p.m. and 11 p.m.
Right. Doing that consistently delayed the physical CC clock by over 3 years.
3 years of youth just from going to bed before 11 p.m. How does that work?
It's deeply tied to our circadian rhythms. Sleeping between 9 and 11 p.m. aligns perfectly with the body's natural peak of melatonin production and the subsequent release of human growth hormone.
Which are the janitors of the cell, right?
Basically, yes. They are critical for cellular repair and clearing out that oxidative rust, the cysteine that we talked about earlier. Miss that window and your cells miss their primary maintenance cycle.
Okay, so that's S for sleep. The W in swag is for walking regularly. Pretty self-explanatory, keeping the motor clock engaged.
Exactly. Physical movement maintains structural capacity.
The A is for apples, which represents eating fruit.
Right, because fruit provides a high volume of polyphenols and antioxidants. These are chemical compounds that directly neutralize that oxidative exhaust. They act as a rust inhibitor.
Rust inhibitor, I like that. And finally, the G in swag. This blew my mind. It stands for grandkids.
Yeah, this was one of the most surprising social findings. Older adults who actively provided care for their grandchildren exhibited significantly slower aging across multiple molecular clocks.
Why? Is it just keeping them busy?
It's biological. Caregiving triggers the release of oxytocin and regulates cortisol, the stress hormone. Lowering chronic cortisol directly reduces systemic inflammation.
So social purpose and caregiving are literally anti-aging drugs.
They absolutely are.
Okay, recap 4. Bullet 1. Your daily habits tangibly change the biological age of your organs.
And bullet 2. Using the swag habits, sleeping before 11 p.m., walking, apples, and grandkids or social caregiving are massive, data-backed anti-aging hacks.
So look at your own lifestyle through that SWAG acronym. Which area is currently aging you the fastest? Are you missing the sleep window?
It's wild to think you're actively turning the dials on your molecular clocks every single day.
It really is. But okay, let's get to the most groundbreaking finding of the entire XAge project. The plot twist.
The driver of aging.
Right. Because up until now, we've talked about these clocks as things that measure aging, a readout on a dashboard. But the data revealed a bodily system that actively causes aging.
Specifically, it hits hard at that second aging cliff in your 60s. We mentioned a massive accumulation of blood coagulation factors.
Right. Factors F5, F7, F9, and one specifically called F13B. Now, coagulation is blood clotting. It's the emergency patch kit for vascular injury. It stops you from bleeding out when you get a cut.
Yes, normally.
So why on earth is a localized repair mechanism causing a systemic aging crisis?
Because as we age, it stops being localized. What the researchers discovered is that F13B doesn't just stay in the bloodstream to form clots. In older bodies, the tissue barriers weaken, and F13B leaks out.
It leaks into the surrounding tissue.
Yes, and once it's outside the bloodstream, it acts as a massive inflammatory trigger. It actively induces cellular senescence.
Senescence. Okay, let's explain that. That's when healthy dividing cells permanently stop dividing, right? They become zombie cells.
Exactly. The leaked F13B forces them into the zombie state. They don't die and clear out like they're supposed to. They just sit in the tissue, secreting toxic inflammatory chemicals that damage all the healthy cells around them.
So the body's emergency patch kit gets stuck in the on position, and instead of fixing a flat tire, it just fills the whole car with expanding foam until the engine chokes.
That is a perfect analogy. But it gets even crazier.
How does it get crazier than zombie cells?
Because the F13B protein also interacts directly with our DNA.
Yeah.
Specifically, it wakes up endogenous retroviruses.
Whoa, wait, wakes up ancient viruses in our DNA?
Yes. Roughly 8% of the human genome is actually composed of ancient viral DNA.
8%?
Yeah, remnants of retroviruses that infected our evolutionary ancestors millions of years ago. Normally, these viral fragments are permanently silenced. They have those restrictive methylation do not read post-it notes firmly attached to them.
Okay, so the cells know not to read the viral recipes.
Right. But F13B acts like a biochemical solvent.
It rips the post-it notes off.
Mechanically, yes, it does exactly that. The presence of F13B in the tissue removes the epigenetic suppression. It wakes up these ancient viral fragments.
That is terrifying. What happens when they wake up?
The cell suddenly starts transcribing ancient viral RNA, and that sparks a massive systemic immune panic. The body thinks it is currently under a severe viral attack.
So the immune system goes to war against a ghost.
Exactly. And that triggers chronic systemic inflammation that rapidly, aggressively ages the surrounding organs.
Mind blown. Okay, so if a blood clotting factor like F13B is the villain actively driving us off that second cliff, does that mean, I don't know, simple blood thinners are the fountain of youth? Should we all just pop anti-clotting meds?
No, absolutely not. Traditional blood thinners are a blunt instrument.
Right, because if you suppress the coagulation cascade globally, you lose that essential repair mechanism.
Exactly. You could easily bleed to death from a minor internal injury. The goal isn't to stop the blood from clotting when you get a cut.
So what is the goal?
Targeted intervention. Preventing F13B from linking into the tissues in the first place, or neutralizing it once it's there without turning off the normal clotting process.
Okay, let's do our final recap because that was a lot of heavy, amazing science.
Let's do it.
Bullet 1. Blood clotting factors, specifically F13B, build up and leak into our tissues as we hit our 60s.
And bullet 2. These leaked factors actively cause surrounding cells to turn into inflammatory zombies, and they wake up ancient viral DNA, causing our immune system to attack our own organs.
So, listener, knowing that your body's own repair mechanisms might eventually turn against you, how does that change the way you view the concept of natural aging? It's not a passive fading away anymore, is it?
No, it's an active, aggressive biological process. And because it's active, driven by specific proteins, it means we can potentially interrupt it.
Right. The precision of the XAge project allows us to move from just mapping the clock to eventually stopping the clock.
Exactly. We are entering the era of geroprotectives. Medications designed specifically to clear out senescent zombie cells and safely neutralize leaked F13B.
Aging is no longer this vague, inevitable decline. Thanks to billions of data points, we know it's a detailed, measurable, and asynchronous process.
We finally have a high-definition X-ray of the aging process.
Which leaves us with a final provocative thought for you to mull over until our next deep dive.
Imagine it's 5 years from now.
You walk into a clinic and a doctor runs your multimodal clock data. They look you in the eye and say, your liver is about to hit the age 40 cliff, or your tissue is starting to accumulate F13B.
And they offer you a targeted geroprotective medication to pause your biological clock right then and there.
Would you take it? Would you actively intervene to freeze your organs in time? Or is the natural asynchronous decay of the human body something we simply shouldn't tamper with?
It's a profound question.
It really is. Think about it. Keep your swag up, and we'll see you on the next deep dive.